I'm Franklin OKEY-IWOBI, and this is my DNA replication project. So DNA replication takes place in the nucleus for eukaryotic cells and in the nucleoid region in the cytoplasm for prokaryotes. And this takes place during the S phase of the interphase, so before mitosis in a cell and before the cell divides. And it does so that when the cell divides, it has another strand of DNA to pass to the new daughter cell that it makes, since every cell needs a DNA. And the key players for DNA replication are the nitrogen bases, which are adenine, thymine, cytosine, and guanine. And also the phosphate group right here, the blue ones, and the sugar, the deoxyribose sugar right here, which makes the phosphate sugar backbone for DNA. So when DNA line up, they line up so the pairs are it's the complementary base pair rule, where cytosine goes with guanine and adenine goes with thymine. And they line up, when DNA lines up, it lines up anti-parallel, which means that one goes from 5' to 3', while the other goes from 3' to 5'. And what this means is that, so when you look at the sugar of DNA, so the backbone sugar, so this is the sugar shape, the oxygen molecules right here, and the sugar is numbered clockwise, so after the oxygen. And this is the sugar on the left strand, which is the leading strand right here. It goes from 1', 2', 3', 4', and 5'. While on the other side, the sugar shape is opposite, which means it goes from 3' to 5' instead. So right here, it's also clockwise. The sugar starts from here: 1', 2', 3', 4', 5'. These are the carbons on the sugar molecule. And right here, it goes from 3', so 3' up here to 5' down here. This goes from 5' up here to 3' up here. And DNA, when they line up, they don't line up, so they're not exactly this pair, and this strand and this strand isn't the exact same. They're not identical. They line up with the base matched to each other. So it's not the same base on both sides, or different base that go as a pair in the complementary base pair rule. And in DNA replication, there are multiple different enzymes. So at first, the first enzyme in DNA replication is the DNA helicase. And what this enzyme does is that it breaks apart. So held by the nitrogenous pairs are held by these things called, they're weak hydrogen bonds that hold the pairs together. And for DNA to replicate, it has to be split apart, open, and then copied, since DNA replicates semi-conservatively. So the first enzyme in DNA replication is the helicase, which comes in. The helicase comes in and it breaks the weak hydrogen bonds that hold the two strands together right here, and then it takes it apart. So after that happens, these proteins called SSB proteins, they come in here, and then they hold the DNA strands together. They hold them apart so they don't get back together, since the DNA needs to be separate for it to be replicated. And after that happens, another enzyme comes in, and it's called the primase. So before the DNA starts to replicate and make new strands, the primase has to leave a primer in here made of mRNA that tells the next enzyme where to start in the replication process. So after that happens, the first DNA polymerase 1 comes in, and then it takes out this primer here. It takes out the primer and replaces it with DNA, since the DNA, it doesn't need RNA in it. It should have DNA instead of RNA in it. And then after the DNA polymerase 1 takes out the primer, the DNA polymerase 3 comes in, and then it starts to build the next matching nitrogen bases, and starts to build the next strand of DNA right here. And then it builds that together right here and joins them together. And then after that happens, the DNA polymerase 2, it comes in, and then it checks, so it has proofreading ability, and then it checks the DNA strands to make sure there are no mistakes, and if there are any mistake, it fixes it and repairs the mistakes if there are any. So this new strand, it goes from 5' to 3', and since DNA polymerase 3 can only work from 5' to 3', this other strand that goes from 3' to 5', it can't properly replicate it as well, since it has to start from here and go down. In the helicase, it's still unwinding it, so it can't start at the extreme while the helicase is still unwinding it. So it creates these things called Okazaki fragments. So as it replicates, it starts from here and moves down, and then a new primer comes in. So it starts here, moves down, a new primer comes in, and then it goes here, and then it makes a new strand as well, and starts from that and moves down and moves down. So after this happens, there are gaps in the DNA right here, and in the last enzyme that comes in is the ligase. And this ligase, what it does is that it fixes the gaps that are left by the Okazaki fragments, so the gaps in between. It fixes it, since DNA can't have gaps in it and it has to be together. And then the ligase, it fixes the gap by joining the two Okazaki fragments together right here. It goes in here. Sure. And right here. And then it joins these two. Take out the primer. And it joins the two DNA. It joins the two Okazaki fragments together, and it's no longer an Okazaki fragment, since it's now a strand of DNA. So since this strand took so much longer to fix, it's called the lagging strand, since it has to make Okazaki fragments and then have a ligase fix it up at the end. While the other strand, it's called the leading strand, since it was just easy and a straight fix. And at the end, this is it. After it's done with that fragment and then they're both done, we end up getting two identical DNA that can go in each daughter cell and have the same exact DNA at the end and make two different cells with the same DNA. Thank you.